Let me welcome everybody to a new global IMMUNOTALK. We're very excited today to have Ruslan Medzhitov, who is going to be introduced by my co-organizer, Elina Soniga. But before we do that, let me just very briefly remind you about the goals of this global IMMUNOTALK series, which are to benefit and inspire immunologists across the world in an egalitarian manner and increase the opportunities to listen to these wonderful talks without traveling. As I said, today we are very grateful to have Ruslan as our global immuno speaker, and I want to draw your attention to our next speaker, Miriam Merat. So stay tuned, and next Wednesday, join us for Miriam's talk. So with this, I'll stop sharing, and I will ask Ruslan to turn on his video, or let's see, maybe we can do, okay, perfect. And he's unmuted, and so Elina, now it's all yours to introduce Ruslan. Thank you.
Perfect. Thank you so much, Carly. It is actually a great pleasure and with honor and also humility, I introduce to you Dr. Ruslan Medzhitov. So Ruslan was born in Uzbekistan when it was still part of the Soviet Union, and he received his bachelor degree from Tashkent State University in Uzbekistan, and then his PhD degree from Moscow State University. In 1994, he started a postdoctoral training in the laboratory of Charles Janeway at Yale University School of Medicine. And in 1999, he became assistant professor at the section of immunobiology in the Yale School of Medicine, where he's now David Wallen's professor of immunobiology and also investigator of the Howard Hughes Medical Institute. So Ruslan has received a number of numerous national and international awards. I really had a hard time selecting only a couple to mention today, so I will mention a few of them. He's Nationally Academy of Sciences in US, and he's member of the National Academy of Sciences in US. He's also fellow of the American Association for the Advancement of Science, and he has received the Shaw Prize in Hong Kong, as well as the Vilsek Foundation Prize in New York, and many, many other honors that he has been awarded. So Ruslan is a giant and undisputed leader in the field of immunobiology. He has greatly contributed to establishing many of the current paradigms in immunology and has really shaped the way we understand and conceptualize the orchestration and regulation of immune response. He's probably best known for his contributions on how the innate immune response, particularly stimulation via toll-like receptors, control adaptive immunity, including the discovery of the first mammalian toll-like receptor, many aspects of toll-like receptor signaling pathways, and several mechanisms of toll-like receptors induced gene expression and antigen presentation. He has also demonstrated the beneficial role of commensal bacteria mediated by toll-like receptor signaling in the intestine, as well as the innate responses to intracellular DNA and the role that endogenous retroelements play in those responses. He made contributions to establish several new concepts in infection biology and tissue homeostasis. I think we will hear much more about that today, including the concept that disease tolerance can act as a defense strategy in mammalian host during an infection. He has also characterized the cellular and organismal metabolism in inflammation, including connecting different effects of fasting, regulating tissue tolerance in bacterial and viral infections. And so we could continue describing Ruslan's contribution for a long while, but I think we are all looking forward to hearing from Ruslan himself. And so I just want to say that it is a great honor to have you as a global immuno speaker today, Ruslan, and we are very grateful for your efforts to prepare this talk and to be here today. Thank you so much.
Thank you very much, Elena, for a very generous introduction, and thank you to you and Carla for inviting me. It's a great thing you're doing for a global community, and it's my pleasure and honor to participate in this.
It is awesome. It's great to have you in the program. And we cannot wait for your talk, but we'll wait a little bit more because we want to ask you a question. As you know, we always ask speakers a question that let us know the speakers better. And I think the audience will be very curious to know what is in your bucket list for your future scientific career. So what's next for Ruslan Medzhitov?
Okay. So the general answer is that I'm really interested in biology that kind of spreads through various borders between disciplines. And what I mean by that is that there are some principles of biology that we can discover within immunology or within neurobiology or developmental biology. But there are some principles you can only discover when you go beyond these boundaries. And that is not easy to do in biology because it's such a vast discipline in general, but that's my main attraction. But more specifically within immunology, what I'm most attracted to is to understand the immune system from the first principles, just the way we understand from first principles, genetics or biochemistry, immunology, neurobiology, developmental biology. I think these are not yet understood from the first principles, and specifically within the immune system, I think we still have a lot of gaps in foundations and a lot of assumptions that need to be continue to be challenged and tested and so on. So that would be my specific aspiration, to try to understand the immune system from the first principles.
Thank you so much, Ruslan, for sharing that. So we will look forward to continue being inspired by your research. Thank you so much. Okay, so we can go on then with your presentation. If you could share the screen.
Okay, can you see it?
Yes, and we can see the cursor as well, so perfect.
Okay, great. So thank you again. This is indeed my pleasure to participate in this seminar series. I slightly changed my title to better reflect what I'm decided to focus on today. I will talk about our recent interest in understanding tissue biology and understanding some basic principles of tissue organization and homeostasis and how it relates to functioning of mammalian tissues and so on, and how these principles evolved over time. So the main question that that guides our sort of curiosity about it is the sort of organizing principles in tissue biology. And what I mean by that is the best way to describe it is by comparison of what we know about organismal biology or cellular biology. So at organismal level, anyone can look at these pictures. These animals look very different, but we know what the common themes are. And these are all mammals. These are all vertebrates. They all have the same body plan. They have the same universal organ systems. And the functional connections between organ systems are well understood. And we can actually even draw a generic vertebrate or generic mammal with all the universal parts. And we also can appreciate that looking at these animals, that though they look dramatically different, these are really variations on that common theme of organization of the mammalian organism. If we now look at different cell types, again, they look dramatically different, and especially if they were shown to scale, some of these neurons would be much, much bigger than some of the other cells shown here. And again, we know what the common themes in cellular organizations are. We know all the organelles, their functions, how they're connected to each other. They're all universal parts. And we can draw a generic cell with a nucleus and mitochondria and so forth. So we know that all these diversity of forms are variations on the theme of a eukaryotic cell. So that's the level of knowledge that we have in the case of cells and all organisms. And now if we go in between at the tissue level, and now we look at these 4 different tissues, and we can ask the same question, are there also common themes in tissue organization? And if so, what are these common themes? And can we draw a generic tissue that would capture all these common themes? What are the universal components of tissues that have to be present in every tissue type? And I think here our knowledge is much more limited. We're not limited by data, by the way, because modern technologies, both imaging and sequencing technologies, allow us to get really unprecedented insight into composition of tissues, into gene expression, and so forth. But that data not necessarily reveals the understanding of this common theme in organization. And that's what we are primarily interested in. So every tissue that part of different organ systems, like shown here, again, look dramatically different, liver, intestine, brain, and so forth. But the reason we think that there are common themes and common features of different tissues is because every tissue has to solve the same set of problems. They have to solve the problem of having appropriate cellular composition, appropriate cell types have to be present. They have to be in appropriate ratios. They have to be in appropriate spatial positioning. This is the same problem that every tissue has to solve. And as biologists, we always can assume, since everything is a product of evolution, once evolution found a solution to some problem, then it just uses the same solution again and again. So it's unlikely that different tissues solve that problem differently. They likely solved it once and early in metazoan evolution, and that's those solutions are the common guiding principles of organization. And tissues not only have to solve the problem of static organization, but also during inflammation, remodeling, development, and so on, composition and ratios of cell types can change. These are, again, the problems that every tissue has to solve. And that's why we think that there are common themes. We think that these common themes are not yet known in the way we know them for cells and organisms, and that's what our main interest is in this area. So the current sort of textbook picture of a tissue would look something like this. So we have many different cell types. We have extracellular matrix. That's really kind of captures some features of the tissues, but it raises many questions. So we have several cell types here. Not uncommon for most tissues to have multiple cell types as their resident types. But the question is, what are the underlying rules to arrive at that are allowed to arrive at this particular composition? What are the relative contributions of different cell types towards tissue organization? We know that if we remove lymphocytes, for example, the tissue would lose some functionalities, but the overall organization of tissue would not be disrupted. However, if we remove fibroblasts, then you cannot really have a tissue. So different cells seem to be have different contribution to overall organization of tissues. And we want to understand what these differences are fundamentally. So in other words, we're asking whether different cell types can be equivalent in terms of their role in tissue organization and function. If so, what are these different types of equivalence groups or cell categories? So cell category would be something that goes beyond cell types. It's a broader category defined by their role in tissue organization. And then what are the relationships between cells that belong to different categories? And so the most primordial type of tissue organization that's found from very early stages of animal evolution, from Placozoa, Spongy, and Cnidaria, we always see this module that's present in all animals. There are no animals that we know of that don't have epithelial mesenchymal module. So we think that this is a primordial sort of primordial unit of tissue organization. And epithelial mesenchymal interactions mediated by various growth factors like FGFs, family, BMP, wing, hedgehog, and so forth. Those interactions really guide animal development and establishment of different patterning systems within tissues. So this is a really primordial way of organizing cells, and generally epithelial cells are functional part of the module, and mesenchymal cells, they play a variety of roles of controlling differentiation of epithelial cells, their function, renewal, and so forth. So these epithelial mesenchymal interactions are connected by various morphogen growth factors. Again, BMP, FGFs, wing, hedgehog, and notch. These are the most universal letters of the alphabet of this communication between these cells during development. And the way that these cells are connected by growth factors is has a lot of meaning. The way that if we draw arrows indicating activation and inhibitory signals, it will form a particular type of a circuit with particular type of topology. And these topologies have very important meaning in these interactions. So, for example, this particular topology where BMP has autoactivation plus activation of epithelial cells, so production of FGF8 that induces inhibitor of BMP. This type of interaction is very famous because this is one example of a Turing interaction that was proposed by Alan Turing over almost 70 years ago, where he proposed that how spontaneous patterns can emerge by having various versions of these interactions where activating signal, for example, can have low diffusion radius and inhibitory large diffusion radius, and then you can have various patterns. And this module can be plugged into proliferation or differentiation, or really into any program. It could be white and black stripes, for example, or it could be anything that it can be plugged in and to generate spatial patterns. So these are very special types of interactions between epithelial and mesenchymal cells mediated by morphogens. And one of our big interests is to try to understand all the different types of circuits that are involved in these interactions mediated by morphogens. The importance of epithelial-mesenchymal interaction has been known for a long time. The seminal experiments by Mangold and Spearman in early 1930s established these types of relationships where dermal mesenchymal cells dictate the fate of epidermal epithelial cells, such that the location of the dermal cells determines what type of appendages are developed by epidermis. In other words, mesenchymal cells dictate the fate of the epithelial cells. And we now know that this is in part mediated by the Hox code, which controls positional information in the mesenchymal cells, and then they dictate different fate choices for epithelial cells. This is a very important principle that I think can be generalized beyond just dermal-epidermal interaction, or even beyond mesenchymal epithelial interaction. Because if we generalize this and think about mesenchymal and epithelial cells as two cell categories now, instead of thinking about cell types, if we think of two cell categories, one cell category, in this case, dermal cells, they have some information. They have Hox gene expression code that determined by positional information, so they contain positional information. And epidermal cells have multiple cell fates, and these mesenchymal cells control that cell fate. And this, we think, can be generalized to say that cells can be divided in these two categories, M cells and E cells. These often are mesenchymal and these often are epithelial, but they don't have to be. The fundamental feature of M cells is that they have some information that's relevant for fate choices by E cells. And E cells have several fate options, but they cannot decide by themselves which one to take, and that choice is dictated by M cells through the signal that they transduce. And these are so-called inductive signals that Spin and Mangold and others describe. So again, I think that we can think of generalized categories of M cells and E cells, not just cell types. And I'll give you a couple of examples illustrating this type of relation between them. So in addition to dermal and epidermal cells, which is the known example, M cells also niche cells would also belong to M cell category, and their partner stem cells would be in E cell category. Because stem cells have several fate options, self-renewal versus differentiation, and niche cells dictate that fate choice. In immunology, we're more familiar with this example. Dendritic cells and naive T cells. Dendritic cells have some information about pathogen, type of pathogen, and so forth. Naive T cells have several fate choices of different effector cell types, for example. And dendritic cells dictate that choice. And when naive T cells became effector cells, now they become M cells because now, upon differentiation into effectors, they have information about the type of pathogen they're specific for. And now when they find their antigen presented by their targets, B cells or macrophages, they will dictate now through signals that they produce, in this case, T cell-derived cytokines, they'll control the fate of B cells for example, isotype switch or fate of macrophages into different M1, M2, and so forth subtypes. So this, I think, is a general principle that cells can be divided into these two categories dictated by this relation, having information versus having choices. So the next question is, what are the possible functional relationships between cell types? So the previous one was about relationship, more about the hierarchy in terms of information processing. And now, what are the possible functional relationships between cell types? And here we have some known, well-appreciated textbook examples of cellular division of labor, where you have two cell types, each specialized on different functions, and that's classical division of labor. Otherwise, these may or may not be functionally connected. Then we have another scenario where two different cell types both contribute to the same function, and they form functional unit, like motor unit. Motor neuron with skeletal muscle would be an example of that. And then finally, we have this type of relationship, which has not been appreciated previously, but we think is very important for understanding tissue organization, and that is relation between a cell type that's specialized on a particular function and another cell type that is specialized on supporting the functional performance of the first cell. So this cell by itself doesn't have a function, but its role is to support this cell. So we call this cell a client cell, and this is an accessory cell or supportive cell by analogy with chaperones and their client proteins. So this type of relation can be illustrated with a couple of examples. So my favorite example would be neuron and Schwann cells. So neurons are obviously client cells. They are responsible for the primary function, which is conduction of the electric potentials. And Schwann cells optimize this function. And without Schwann cells, neurons can operate. But if it's a large neuron, then it will not be able to operate as well without Schwann cells. However, Schwann cells, as far as we know, without neurons, don't really have any function. And the same is true for Sertoli cells and differentiating spermatogonia, where spermatogonia and sperm cells are obviously client cells. That's the purpose of this tissue. But Sertoli cells perform supporting functions. So again, they're accessory cells, and these are client cells. So now if we look again at this picture, now we can look at it from different perspective. We can say, well, all these cells are there, but they're not equal. Some of them have some information that they share with other cells to dictate their fate. Some of them have primary function and some of them have supportive functions. And if we now try to strip it down to what would be the bare essentials, cell types that would be present in every tissue as a minimal unit of tissue organization, at least in vertebrates. If we ask that question, we think that the minimal unit of organization in vertebrates is made up of 4 types of cells. The main one, the cell type that is tissue specific, or for the lack of a better term, we can call them parenchymal cells, but these are really client cells. They are the functional part of the tissue. Typically, it would be, for example, epithelial cell. They're responsible for primary function of the organ, like transport, digestion, absorption, and so forth. And there are always 3 additional types of cells that are not the primary part of the tissue, not the primary function of the tissue, but they perform supportive function. There's always microvascular endothelial cells, obviously, because all tissues need them to deliver oxygen and nutrients and remove waste. There's always some stromal cells like fibroblasts, and there is almost always resident macrophages. So these 3 cell types appear to be universally present in all tissues, and this cell type would be different between different tissues, and this is the functional tissue-specific cell type. It's a client cell, and these are accessory cells. And that is the reason why without these 3 types of cells, tissue cannot really operate normally. It cannot be organized normally. So this we think of as a minimal tissue unit, minimal unit of organization. And how they are assembled within tissues, that becomes a big question. So in order to assemble and persist within tissues, each of these cell types need a specific growth factor. VEGF, for example, for endothelial cells, PDGF for fibroblasts, CSF1 for macrophages, and then the client cells will have different growth factor depending on what cell type they are. But these growth factors have to be there in the right amounts, right proportions in order to establish this unit. All additional cell types, lymphocytes and adipocytes and so forth, that can be present in many tissue types, these are add-ons. These are superimposed on this minimal unit of organization. So the question then becomes how production of growth factors is regulated. And here we need to distinguish between morphogens, these inductive signals, FGFs, BMPs, and so forth, that control common developmental routines such as spatial patterning, polarization, branching morphogenesis, and so forth. That's the one I mentioned earlier in the context of epithelial-mesenchymal interaction. But here we focus on these so-called lineage-restricted growth factors that control differentiation, survival, and proliferation of specific cell types. So this would include CSF1 for macrophages, PDGF, VEGF, IL-5, IL-7, and so forth. So these are all lineage-restricted growth factors. They're necessary for specific cell lineages to stay alive and to proliferate. And there is one big unresolved question in biology, I think, is about control of growth factor production, because all cells, untransformed cells, require specific growth factors. But they need to be produced in appropriate amounts and locations to ensure stable tissue organization and composition. So that's clear. But that means that there are some rules that determine production of this class of growth factors, and these rules are largely unknown. And that's one of the questions we're very interested in, and I'll illustrate it here. If we have 2 cell types, X and Y, typically lineage-restricted growth factor for one cell type is made by another cell type. So here cell X makes growth factor for cell Y, and the amount of this growth factor made by cell X will determine how many cells Y we will have, and what's the ratio of X to Y. And then the question is, how does this cell know how much growth factor to produce to have appropriate numbers of these cells in appropriate ratio? And that was one of the questions we started with. And we started with two of our favorite cell types, fibroblast and macrophage. We could use primary cell types for this experiment, which was necessary. These cells are part of the universal minimal unit, so clearly important part of tissue organization. And we tried to reduce the problem as much as possible to like simplest variables that we could experimentally manipulate in vitro. So first we documented and confirmed that indeed fibroblasts produce CSF1, macrophages express receptor for CSF1, macrophages produce PDGF-beta for fibroblasts express receptor, so there is this part of the loop, and also fibroblasts can make autocrine signals like PDGF-alpha, for example, so there's also this part of the loop. And then we asked whether macrophages and fibroblasts can actually support each other since they make growth factors for each other. Normally, when you culture primary cells, you have to provide growth factors for them to grow. But here we found conditions where there was no exogenous growth factors, and all growth factors have to come from either macrophage or fibroblast. So they have to feed each other. And we found such conditions and found that surprisingly and interestingly, not only they can support each other, but they actually have this very interesting feature that if we mix fibroblasts and macrophages in the absence of exogenous growth factors, they always achieve the same stable ratio, even if we vary by 100-fold the initial ratio between them. Over time, they converge on a stable ratio. So that's not a trivial feature, and just to explain some of the rationale in the subsequent experiments, I need to just remind you that when replicating, self-replicating populations grow, they can enter exponential growth phase, but it's always restricted by something. There is nothing that can go on exponentially. There's always some restriction. In the case of cells, it would be, for example, available space or oxygen or nutrients and so on. And that establishes the ceiling for the growth. And this is what's called carrying capacity of the environment. And that results in a logistic growth mode, which is the sigmoidal function. And the cell number has to be lower than the maximal possible cell number in population for this term to be positive and for cells to grow. If it's larger than K, then they will decline. So there is also an additional version of it when the cells need to have minimal density called allele number in order to undergo logistic growth, and below the allele number they decline, and I'll get back to that later. So carrying capacity is an important concept that comes from ecology, and it's basically any kind of extrinsic limit on population size. It could be due to availability of resources or space. For cells, it could be nutrients, oxygen, growth factors, space limitation, and so forth. Whichever factor is limiting would be carrying capacity factor. And this limitation imposed by carrying capacity converts exponential growth into logistic growth, as I showed in the previous slide. So exponential goes into logistic growth. So at that point, we started a very nice collaboration with Uri Alon and his student Miri Adler at Weizmann Institute. And what Miri did, she looked at, since there is always has to be extrinsic limitation on growth, we assume that there's always going to be some carrying, at least one cell type in the circuit has to be limited by carrying capacity. And then there are multiple ways that these two cells can exchange growth factors. And if we look at all possible ways you can draw this circuit, there are 144 possible versions. And Miri did then this heroic analytic screen, solving systems of differential equations to find which of the circuits would be stable. And what she found is that out of 144, only 24 circuits were predicted to be stable based on this analysis. And then she asked the 24 circuits that are stable, do they have some shared features? And very satisfyingly, there were just two shared features, and they're shown here. What the shared requirements are that one cell has to be limited by carrying capacity, by external limitation. The other cell type doesn't have to be, but it has to be limited by negative feedback on the growth factor produced by first cell. And the growth factor, and that's negative feedback, there are two versions of how it can work, and that's why there are two possibilities listed here. So that was very interesting that there are, in order to have a stable two-cell circuit, all you need is to satisfy these two requirements. And so these requirements would be limitation by carrying capacity on one cell type and negative feedback on growth factor produced by that cell type. And so these are some examples of stable and unstable circuits. And this is the one we observed with macrophage fibroblasts. This is the one, these two would also be stable, but probably some other cell types may have these circuits. And this would be unstable. It doesn't have stable ratio between X1 and X2, between two cell types. And you can notice here that in the observed circuit, fibroblast macrophage circuit, in addition to this stable state with stable ratio between the two cell types, there is another stable state where you just have fibroblasts without macrophages. But in order to achieve this stable state, you need to have critical density of fibroblasts. That's what Ali effect is. And we think that this creates a vulnerability in the system so that now a system like this can be vulnerable to transition to a state dominated by one cell type. And that's exactly what may be happening in fibrosis. So fibrosis might be possible because of this. And the reason this is possible is because of the autocrine signal here. So here in this case, in both cases, you have this alternative stable state. So we think fibrosis is enabled by this vulnerability of the system. So this two-cell circuit then have these features, two types of restrictions, either by space on one cell type or by negative feedback by another cell type. And they have these characteristics. There's chief stable ratio, they are robust to perturbations. This is minimal circuitry that's necessary. And we think that can be potentially expanded to other two-cell circuits, including DCs or T cells, neurons, and so on. The growth factor would be different in each case, but it's likely to follow the some variation on these rules. So having found this feature for establishing stable cell ratios, we next asked the question, how cell numbers within a given compartment are controlled. Each tissue compartment has appropriate number of cells of different types. And the question is, how is that regulated? And that question, of course, is a very old question, and people have been thinking about it for a long time. And surprisingly, there are actually two paradigms to answer that question that coexisted. They're alternatives. They're not, they're a different way to answer the question, but they coexisted for a long time. The first paradigm is space availability. So very simply, so cells will grow until they fill up all the available space. And this is what's observed in vitro in the phenomenon of contact inhibition. If you grow fibroblasts at some point, when they become confluent, they stop growing. So that paradigm certainly true. It existed for a long time, and it's true for cells like fibroblasts and epithelial cells. But you can imagine for hematopoietic cells that wouldn't work because hematopoietic cells are not limited by space. And then there is the second paradigm, which was proposed by Martin Ruff in 1985, initially, I think, where the cell numbers are controlled by growth factor availability. So you have a certain amount of growth factor, which is predetermined genetically, for example, in a given location. And then the cells will proliferate until they use up the growth factor. So the amount of growth factor then will control how many cells you will have per compartment. And this paradigm works well for hematopoietic cells, like macrophages and lymphocytes. And we know that if you increase growth factor concentration, you can have more lymphocytes or more macrophages. So there are two paradigms. And what was striking about it, we thought that, well, this really represents two paradigms that relate to the two cell types we studied in our circuit. And indeed, if we look at fibroblasts, they do follow growth control that is dependent on cell density. Once they fill up this space, they stop growing. So they're very density dependent, their growth, but not growth factor dependent. Whereas if you take macrophages, they have low sensitivity to density. And this is actually reflection of their growth factor depletion. So they're not really limited by density at all, but they're very much sensitive to growth factor concentration. So we have two cell types within the same circuit that represent examples of the two different paradigms I just mentioned. So one cell type, fibroblasts, limited by space. So that would be carrying capacity limitation. Another cell type, macrophage, is limited by growth factor produced by the first cell type. So now within the circuit, we have both paradigms. And then we thought, well, perhaps indeed, this is not a coincidence that within this circuit, stable two-cell circuit, remember I mentioned that you always have the requirement for extrinsic limitation, for example, by space. But you only have to have it on one type of cell within the circuit. The other cell type doesn't have to be limited by carrying capacity. And so we then thought, well, okay, fibroblasts follow these paradigms, space availability, macrophages follow this paradigm for growth factor availability. And then, of course, the next question was whether these two paradigms are actually connected, such that production of growth factor by fibroblasts is a function of space availability for fibroblasts. So fibroblasts would sense space availability, and that would translate into the amount of growth factor made by cell type that is not limited by space availability, and that would be a beautiful solution of this problem. So we addressed that in a variety of ways. I will just summarize it quickly here. So first we found that fibroblasts do exhibit density-dependent gene expression program. So these are different densities of fibroblasts, and there are multiple gene categories depending on how they control by cell density. And what was satisfying is that there are many growth factors that we found to be regulated in a density-dependent manner, including CSF1, which was suppressed at high density and induced at low density, as shown here. And when we looked at the genes carefully, at the types of pathways that regulate them, these three pathways came up as the most dominant. It's a HIPPO signaling pathway, TGF-beta pathway, and Wnt pathway. And this is based on enrichment for binding sites and genes that are controlled under these different density conditions. And so this is an example of TGF-beta pathway being controlled by density. So this phospho-SMAD2 and 3 activated low density, suppressed at high density. And the HIPPO pathway example is shown here, where at low density, HIPPO pathway is off, YAP is nuclear and induces gene regulation, whereas at high cell density, YAP is excluded from the nucleus and the transcription is turned off. So it is regulated by cell density as expected. And what we found is that YAP indeed regulates CSF1 expression. This is constitutively active YAP in fibroblasts, but not other growth factors that are density dependent. TGF-beta, on the other hand, doesn't control CSF, but controls these other growth factors that are density dependent, and then Wnt controls yet another subset of growth factors. So each of them regulate different subsets of growth factors in a density-dependent manner. And in the case of CSF1, this was indeed not dependent on TGF-beta, shown here as inhibitor or knockout of the TGF-beta receptor, whereas these other growth factors were TGF-beta dependent. And when we put this constitutively active YAP into fibroblasts, what we found is that it induces CSF1 expression, and it alters the numbers of macrophages and fibroblasts as would be expected, and it changes the ratio, stable ratio between macrophages and fibroblasts. So they still achieve stable ratio, but now it's a different set point. And this is what's shown here. This is wild-type cells. So this is to remind you that they achieve stable ratio. This example is wild types. They achieve the stable ratio starting with different initial ratios. And when fibroblasts express constitutively active YAP, increases CSF1 production, there are more macrophages, macrophages make more PDGF beta, and you achieve stable ratio at a different value of these cell types. And what was interesting also is that this HIPPO pathway dependence of CSF1 production by fibroblasts appears to be restricted to certain cell types. So looking here at the control regions of CSF1 gene in different cell types, you see that there is an active enhancer determined by H3K27 acetylation, active distal enhancer 10 kb away from the gene, that is only present in fibroblasts but not endothelial cells or leukemia cell line. And this is chromatin IP experiment showing that YAP indeed is recruited to this enhancer in fibroblasts. So this enhancer provides fibroblasts with this unique sensitivity to the with the unique coupling of the space availability, which is sensed by HIPPO pathway, with the induction of CSF1 necessary to support macrophages. So fibroblasts exhibit density-dependent gene expression, including control of various lineage-restricted growth factors. I focus on CSF1 as an example, but as I said, there are multiple growth factors that are regulated in this manner for other types of cells beyond macrophages. And HIPPO pathway and TGF-beta signaling regulate these density-dependent gene expression programs, along with Wnt pathway. And YAP controls CSF1 expression in a cell type-specific manner through this cell type-specific enhancer. And so sensing of the environment by fibroblasts, therefore, is linked to the control of population size of macrophages via control of CSF1 expression. So one cell type senses space availability, that sensing converted into control of growth factor for the other cell type that is not limited by space. And in this way, this circuit solves the problem of having the appropriate cell composition and ratio. So here, if you have low fibroblast density, it activates YAP, leads to CSF1 production, macrophage expansion, increased PDGF beta, source fibroblast expansion, and the circuit is closed. And fibroblasts would expand only until they are limited by carrying capacity, like space limitation. So this can be further expanded now. The example I showed you, the space as a limiting factor of carrying capacity, sensed by HIPPO pathway, controlling CSF1, translating in the appropriate macrophage cell numbers. But in other scenarios, it could be other environmental factors that can be limiting. For example, oxygen would be sensed by HIF pathway, translated to another growth factor, VEGF, and that would control proliferation of endothelial cells. So it's the same principle, different growth factors, different cell types, and that of course controls homeostasis of oxygenation within tissues. And then some unknown factor, we don't know yet what that is, that through TGF-beta pathway controls some other growth factors. We know several examples of these growth factors that are controlled by TGF-beta, but what's upstream, we don't know yet. But it also is density dependent, but distinct from this one. So in other words, sensing of the environment by one cell type, typically stromal, mesenchymal type of cell, like fibroblasts, is linked to the control of population size of another cell type that is not limited by carrying capacity. That's the general principle, we think. And under conditions of inflammation, some of this control can be overtaken, for example, by cytokines that through different pathways, through NF-kappa B, can control CSF1, to expand macrophages in monocytes in a sort of on demand when there is a, so it's overrides the homeostatic control and increases them in the case of infection, for example. And putting it together with other cell types, this minimal unit of tissue organization, we think all of them are connected by control of growth factor production by sensing some kind of a functional demand for oxygen, for extracellular matrix, or for whatever function is performed by the client cell. And this way they maintain appropriate, adjustable, and flexible composition of the different cell types within the tissue. And in the final minute, I just want to relate this to the idea of tissue homeostasis. So any homeostasis is all about having some variable that you care about, something you want to regulate, such as oxygen, glucose, and so forth. There must be a sensor that can monitor the value of this variable, and there must be an effector that can change that value in the desired direction, and then some signal that connects sensor and effector. So in the systemic homeostasis, let's say blood glucose is sensed by pancreatic alpha and beta cells. If glucose is low, alpha cells will make glucagon that will induce glycogenolysis, gluconeogenesis, and hepatocytes release glucose and raise glucose levels. So these always operate by negative feedback to correct the value of deviation of the regulated variable. So systemic homeostasis is well understood, cellular homeostasis is well understood, but tissue homeostasis is not as well understood because we often don't know what the regulated variable is. With a few exceptions like hypoxia. Hypoxia is a well understood example, of course. Sensor cells, here, by the way, in systemic homeostasis would be endocrine cells. In the tissue homeostasis would be macrophages, so they're tissue equivalent of endocrine cells. They detect deviation in oxygenation, in oxygen concentration. If it's low, produce VEGF, induce angiogenesis to correct it. So this is one example of regulated variable within tissues, but what we really need to know to understand tissue homeostasis, we need to define all regulated variables. What is being maintained by tissue homeostasis? And again, other than oxygen, it's largely unknown, but these are some likely examples of regulated variables within tissues. This would be oxygen and nutrients. How are they corrected if they deviate? Through local blood perfusion and angiogenesis. Another regulated variable, cell number and composition, that's the one I focused on today. This is controlled through cell proliferation, cell death, and cell migration. That's the way to change cell number and composition. ECM, very important one, and they're probably least understood in terms of how it's regulated, its composition, mechanical properties. But fibroblasts and macrophages are involved in controlling ECM homeostasis. Fibroblasts by producing ECM and macrophages by degrading it, for example. And then there are interstitial volume, pH, osmolarity, metabolic waste products. And again, these are various candidate processes that can control them. And ultimately, there is the sensor cell, like cell X, always determines the values of regulated variables that can be changed by cell Y. So if there is deviation, it's a signal to cell X that there is a demand for function performed by cell Y. And that would translate into growth factor production for cell Y to expand it so it can perform that function. That's a homeostatic circuit that couples what I described for growth factor control with tissue homeostasis. And another version of it, of course, would be, it's not necessarily expanding them, but it could be recruiting them, like in inflammation, or activating them to increase this functional performance. And that's how we think this growth factor production is coupled with homeostasis. And I'll stop here. So the work was primarily done by two very talented postdocs, Ruth Franklin and Xu Zhao. Ruth already just left the lab to set up her lab, her own lab at Harvard, and Xu will follow very soon as well to establish his own lab. And Matt Medzhitov and Scott Popp helped with various aspects of the project. And again, this was really, the first part was entirely collaboration with Miri and Ria Lon from Weizmann Institute, and this is our support. And thank you very much for giving me a chance to talk to you, and thank you for your attention. And thank you, Elena and Carl, again.
Thank you, Ruslan. That was wonderful. Thank you, especially for sharing your discoveries on the fundamental principles that drive this tissue organization, and in particular for sharing how you approach this conceptually. I think this is a true inspiration for all of us. So
thank you. Thank you.
Very grateful. So before we leave, I just want to remind you on how we're going to ask questions that I'm sure you have for Ruslan. So as always, we're going to do this through Twitter. So there's already a tweet that you can find that says, ask questions for Dr. Ruslan Medzhitov here. Just remember to add the hashtag #GlobalImmuno. And Ruslan have recently gone into Twitter, I understand, and he will be answering his questions through his account that is at @Medzhitov. So thank you so much, Ruslan. And yes, so we'll see all of you next week when we'll have Mira Murad joining us. See you.
Thank you. Thank you, Ruslan, again, and fantastic seminar, thought-provoking as always. Thank you so much.
Thank you very much. Thank you. It's my pleasure.
Bye, everyone. See you next week.
Bye. Bye.